Hello and welcome to part 2 of Chapter 4, a tour of the cell. I'm Dr. Chris Whips. Let's get started. So as we make our way around the cell here, we're going to now talk about mitochondria and chloroplasts. And I show this image, again, just as a reminder here, plants being eukaryotes, they do have mitochondria. And then, of course, plants also have chloroplasts, and let's get into both of these now. We'll start off with the mitochondria, and it's helpful to take note of some of the anatomy of a mitochondrion. So here we have the mitochondrion. It has a double membrane outer layer here, so that's going to be important. We'll come back to that. So there's an outer membrane and inner membrane, and that inner membrane is highly convoluted or folded. And we'll see how that's really important for providing a lot of surface area when we get into metabolism. Mitochondria, and as we'll see, chloroplasts, have their own ribosomes. So those protein building machines, they have them, and they're of mitochondrial origin. They also have their own DNA. So mitochondria and chloroplasts have their own genetic information. These infoldings are called cristae, and that cytoplasm you can almost think of inside of this inner membrane is called the matrix. So there's your mitochondrial anatomy. What do mitochondria do? They are the site of cellular respiration, and that's what we'll spend some time on in some upcoming chapters. We'll be looking at what exactly is happening in those mitochondria. Mitochondria are kind of interesting in the sense that we see these cartoons of them as sort of these little bean-shaped looking things, but actually they can elongate and extend. We see here in this picture of, this is a euglena. This is a free-living single-cell protozoan eukaryote. And notice you don't see little beans in here, but kind of these elongate structures, and they actually maybe interconnect with one another. The mitochondrial DNA here is staining orange, these little dots. But notice these almost tubular-like mitochondria. Here's a transmission electron micrograph showing how much more elongate a mitochondrion actually might appear in reality versus, again, this sort of cartoony bean-looking thing. All right, chloroplasts now. This is the site of photosynthesis. So in plants, this is where they harvest that sunlight, energy from sunlight, and convert it to chemical energy. That's happening here in the chloroplast. We'll get into a lot of that more detail when we talk about photosynthesis in a few chapters from now. But for now, let's look at the anatomy of a chloroplast. They have an outer and inner membrane like we saw with mitochondria, so this double membrane structure. But chloroplasts also have these additional membranous structures, compartments within that inner membrane area, and these are called thylakoids, and therefore the actual membrane around these is called the thylakoid membrane. That's going to be important when we get into photosynthesis. So it creates this compartmentalization and really these microenvironments within the thylakoids. That'll be important. What we would think of as cytoplasm within this inner membrane area would be, it's called the stroma. The thylakoids have the thylakoid membranes, and then inside of them, the thylakoid space. It's not shown on here, but that's what that's called. We also, again, see ribosomes, these little dots, and chloroplasts have their own DNA. So these are interesting things, and this feeds directly into this next concept as follows. The idea of endosymbiosis. So endo means within, symbiosis means living together. And mitochondria and chloroplasts, we have pretty solid evidence at this point that they arose through an endosymbiotic event. So what does that mean? What happened? So if we look back through evolutionary time here, we have some proto-eukaryote. So first there are prokaryotes. Those occur before eukaryotes. We see the origin of a nucleus. How that developed is a little more open to speculation. But you have these membranous compartments. So we're starting to see something that looks more like a eukaryote where you have compartmentalization. And then at some point, we see an uptake of an aerobic prokaryote. So these are probably living in association for a very long time. Eventually, this bacterium, prokaryote, gets taken up into that proto-eukaryote, and that association becomes permanent. Again, over time, this isn't just like a one-time event, but over time that association becomes permanent, and then over further time, that the mitochondria is going to lose its ability to be a free-living thing completely, and this association becomes permanent. Later on in plants, we see the acquisition of a chloroplast, which is a photosynthetic cyanobacterium. That's the origin of those. So what is the evidence for this? So there's a few things to pay attention to. Remember, mitochondria and chloroplasts, they have their own DNA. So if we look at that DNA, it is of prokaryotic origin. They have that double membrane structure. So when we think of these cells being initially phagocytosed, you'd have the membrane encircling that phagocytosed prokaryote. And so that would lead to a double membrane structure. So that's important. These guys, they look like a prokaryote. So if we look at the size of them, they fit in terms of the size. Chloroplasts and mitochondria divide through binary fission. That is how prokaryotes divide. Ribosomes, I already mentioned that. And so these are the bits of evidence that we have. If we look a little deeper, we actually go and look at the genetic material of these organelles. We can put them on a phylogenetic tree. And what we see is those lineages, the genetically, mitochondria and chloroplasts fall into the bacteria. Mitochondria are proteobacteria, chloroplasts were cyanobacteria. And so we can kind of draw lines connecting where this association, this endosymbiotic association, became permanent with the uptake of, say, mitochondria, or what was a protobacterium, or cyanobacterium here in plants, in the evolution of eukaryotes. Really cool stuff. Another organelle that's related to metabolism is called a peroxisome. And these are associated with oxidation. They have many functions, so they are important for breaking down long-chain fatty acids. So again, we're thinking from a metabolic perspective, breaking down fats, right? So that's important. They are important in detoxification. So if you look at what, say, a liver cell does, and in this example, related to alcohol, these will break down about 25% of that alcohol. A lot of the other detoxification happens within that, think back, smooth endoplasmic reticulum, right? The origin of peroxisomes, evolutionarily speaking, is not really known, not really understood. So not necessarily through like an endosymbiotic event like mitochondria or chloroplasts. But they're often associated with mitochondria and chloroplasts. So we see in this TEM here, here's a peroxisome, and there's chloroplasts on either side, and here's a bit of a slice of a mitochondrion here as well. And so what these guys do is they produce hydrogen peroxide, H2O2. They take hydrogens from other compounds and add them to oxygen. And so that's how they produce that hydrogen peroxide. Then they convert that to water. Sort of maybe in some ways sounds convoluted, but that's how they work. And that's why they're called peroxisomes, because of this peroxide. All right, quick review. Where would you find DNA in a plant cell? Answer this question. All right, yes, so given the choices, you would find DNA in the nucleus, chloroplast, and mitochondria. Again, this is just reiterating this idea. I think where the misconception comes in sometimes is that plants have chloroplasts, but not mitochondria. But they're eukaryotes, they have both. Let's talk now about the cytoskeleton in cells. Here's a really lovely picture. These are cells from a cell culture. What you're seeing here, the blue dots, those are the nuclei of cells, this monolayer of cells. And then you have these cytoskeletal elements. There's a couple of different kinds here, stained with different colors. And so that's what you're seeing with the green and red, these different kinds of elements that help give cells their structure. So here are the different kinds of cytoskeletal elements, actin filaments, microtubules, and these intermediate filaments. We'll go through each one in turn. But what's the purpose of this cytoskeleton? This provides structure for the organelles that we've already talked about. It kind of keeps them in place. We see in this little cartoon here, this mitochondrion is actually connected to the cytoskeletal network and how it maybe moves through that. It's important what the framework is around all this. So the cytoskeleton also makes the shape of the cell. That's particularly important for animal cells. Plant cells are different because they have that cell wall around them. Moving materials in cells, not just, shifting vesicles around and moving them from one organelle to the other or moving something to the periphery of the cell to be secreted. But when we get into cell division, these components are going to be really important for moving chromosomes around, making sure those get to the right places in their daughter cells. All right, so here are these three elements just looked at in a different way: microtubules, microfilaments, intermediate filaments. I like this little cartoon. It gives you a little bit of a sense of where some of these things occur. This is meant to be like intestinal epithelial cells. So here's tubulin or microtubules made of tubulin radiating out from this area called the centrosome. Here are microfilaments providing kind of the skeleton or shape around the periphery of this cell. And here are these intermediate filaments really providing strength to maintain cell shape. That's part of it, but also interconnection with other cells. So let's talk about these microtubules first. They're tubules and they're hollow. So they're made out of this protein called tubulin. You get two different forms of tubulin, alpha and beta tubulin, and those come together in a dimer. So that's made of two parts, and those are added one at a time onto, say, a growing tubule on one end. So that would be this polymerization step. These are sort of charged with GTP. Remember, ATP being the currency of the cell. This is, I call it the Canadian currency of the cell, GTP. And so these are added on one end as charged tubulin dimers. That's polymerization. But they can be broken down easily too. And that'll be important when we think about how dynamic this is and how quickly cell shape sometimes has to change or material get moved around quickly. And that's through depolymerization. So they can break down fairly quickly. There are several important structures in your cells that are made out of microtubules. So the first one here are the centrosomes and centrioles. So in animal cells, like our cells, microtubules will radiate out from these locations. So the centrosome, which is near the nucleus, that's kind of it's also called a microtubule organizing center, MTOC. That's where microtubules will grow from. And they themselves, these centers, are made out of microtubules. So we see them here. We have little clusters of microtubules. Here's three of them, three. We're seeing cross section here. So they have this 9x3 arrangement. And just to clarify, the centrosome is made out of a pair of centrioles. So each one of these structures is a centriole. That makes up the centrosome, which is this organizing center. Another place that you see microtubules are in flagella and cilia. And flagella and cilia are important for movement of eukaryotes. So cilia tend to be shorter and move in sort of a beating-like fashion, where flagella kind of whip around and undulate. But yes, they're made out of microtubules. Here's what that looks like. It looks similar to those centrioles. You'll see these here. So here's a basal body, basically sort of the root of a flagellum or cilia. This looks a lot like a centriole. It has these 9 groups of these triplets of microtubules. When you get a little further out in a flagellum or cilium, what you see are some central microtubules as well. And so this is also known as this 9+2 arrangement, where you have these two additional microtubules, 9 around the outside, 9 sets around the outside, and then 2 in the middle. All right, quick review. What protein do you need to make flagella and cilia? So you got to think back. What are these made out of generally, and what is that thing made out of? All right, so yeah, given the choices, they are made out of tubulin. That's what microtubules are made out of, conveniently named tubulin. Now, I bring it up here to really make this contrasting point. You also find flagella on prokaryotes, but these have a different evolutionary origin. These are not made out of tubulin. You do not find microtubules in prokaryotes. So prokaryotes have a cell wall that kind of gives their cells strength and structure. They don't have that internal network like we see in eukaryotes. Flagella in prokaryotes are made out of a different kind of protein. How the flagella work in prokaryotes is different. We see a little cartoon here of the flagellum really just rotating as opposed to moving in a whip-like fashion. And so we can contrast these two things. So prokaryotic flagella are made out of flagellin. Again, that's kind of a convenient name. Eukaryotes, it's made out of tubulin because it's made out of microtubules. The prokaryotic flagellum is thinner, less complex. The energy source for these with prokaryotes is a proton gradient. With eukaryotes, the energy is derived directly from ATP, whereas here the proton gradient is an indirect way to get that energy. But back to our microtubules, another function of microtubules within the cell is actually to act as little highways and move things around. So not just through polymerization and depolymerization, but having a structure for other proteins to move along. An example here would be this motor protein. One is called kinesin, kinesis, it means movement. And you can check out one of these videos here. There's some really cool animations showing this. These little almost legs of this kinesin protein will move. One will step forward, that uses one ATP molecule. And then it'll kind of walk along this microtubule. And in this case, it's moving or dragging along this vesicle to take from one place in the cell to the other. So microtubules can also act as these highways. All right, how about microfilaments? So we're not talking about tubules now, we're talking about filaments, and they are not tubular, so there's no hollow middle. So these are thin rods. They are made out of actin. That's the protein you see in microfilaments. You probably heard of actin when you think of like actin-myosin interactions in muscle cells, and we actually will see that here, actin and myosin interacting and being important in how cells might move. So these are important around the periphery of the cell, kind of bear tension, give that cell, animal cells in particular, strength. We see a lot of actin in the microvilli of intestinal cells, so we see that here. So you have these villi projecting out from these intestinal epithelial cells. You also get bands of actin filaments helping connect these two cells. That's going to provide strength of that layer of cells. But also you see them extending out in red here and being cross-linked by other proteins, giving that microvillus that structure and its strength. Now remember, actin-myosin interactions, you might think of muscle contraction, but that same thing can be happening within actin filaments inside of other cells. So the movement of myosin along those actin filaments might be important within a cell. Actin filaments polymerizing, extending, getting longer is going to be important for extending these like pseudopodia in an amoeba, or here's a macrophage grabbing up little bacteria. So as one of these pseudopodia extends, that actin filament is getting bigger, but it's depolymerizing on the other end. And so this kind of gets to the dynamic nature of these kinds of cytoskeletal structures, is they can quickly get longer, maybe on one end, and be depolymerizing on the other end. So we see that with both microtubules and microfilaments. So this can happen in a very dynamic way. And if you go back to the previous slide and watch one of those videos of a cell moving, there's this great one of a neutrophil chasing two bacteria, and they eventually gets it. It's all happening in real time and very quickly. And all that movement of that cell that you're seeing under the microscope is microfilament polymerization and depolymerization. All right, our third kind of cytoskeletal structure, these intermediate filaments. So they're kind of, well, intermediate. They're bigger than microfilaments, but they're not tubular like the microtubules. These are important for keeping organelles in place, providing structural support for tissue layers. So we'll see how there are actually connections between cells with these. And we can look at some images showing this. So here are intermediate filaments composed of keratin. You probably might have heard of keratin. Staining red here. There are other intermediate filaments, like these ones composed of lamins, that are important for the structural support of a nucleus. So there's different kinds of intermediate filaments. Here we start to see those connections between cells. So here's a layer of skin cells here, and you see the green intermediate filaments really almost, there are these junctions that hold the cells together, and those intermediate filaments connect to those points. And so this is providing a lot of strength for that tissue layer. All right, let's work our way outside of the cell now and talk about extracellular structures. So extracellular means outside of the cell. And so whether we're talking about plant or animal cells, we see cells produce materials that get exported, but then aren't just like secreted out into the milieu and disappear, but they actually form part of the structure around the cell. All right, so let's talk about first the cell walls of plants. So this is an extracellular structure, and this is something you don't see in animal cells. And a cell wall provides protection and shape, and important for sort of resisting the loss of water, but also preventing the excessive uptake of water if the plant is exposed to water, as we'll get into when we talk about osmosis here down the road. So the plant cell wall is made of cellulose. Remember that structural carbohydrate that we find in plants. There's other polysaccharides and proteins that are associated with the cell wall too, but largely this is cellulose. There are potentially 3 layers to the cell wall. The primary cell wall, that's shown here in the pink or red color here. There's this middle lamellar area. You can really see it more clearly in this zoomed in and colorized transmission electron micrograph. So that's the area between the cells. And then in some cells, like when we think of like wood, we also see this secondary cell wall between the plasma membrane and the actual sort of contents of the plant cell and that primary cell wall. In animal cells, they don't have cell walls, but they do have this thing called the extracellular matrix. So this is made of proteins and glycoproteins. So a glycoprotein is a protein with carbohydrate groups attached to it. Collagen would be an important component of the extracellular matrix. That matrix itself may be connected to a cell, to the cell membrane, by a protein called integrin. And that one will come up again in a bit. If we look at a tissue, like a connective tissue, here we see cells in this cartoon version here. The cells themselves only make up part of it, but really a lot of the connective tissue is made up of this extracellular matrix stuff, elastin, collagen, for example, and then this sort of gel-like material in between. So collagen is a very common protein. This makes up 25 to 35% of your total protein, period. So this is like of all the proteins in your cells, this is 25% plus of your protein. You find it in bone, cartilage, skin, basal lamella. We'll show you what that is in a second. But collagen fibers form these like cable-like structures here. So this provides a lot of potential strength. So let's see. Yeah, basal lamella. So what is a basal lamella? This is the basement of these epithelial layers, for example. So here's an intestinal epithelial layer. It's one cell thick. And at the basement or bottom of it is this lamellar area. And this provides a connection to this connective tissue. So basal lamina and the connective tissue. And this is going to provide that tissue layer with additional strength and structure. And that's particularly important in intestinal tissue because this is only one cell layer thick. Okay, quiz time. How are cells connected to the extracellular matrix, like the basal lamina? What protein is involved in that connection, given the choices you have here? All right, so you might have remembered from a couple slides back, or you could have just looked it up in your textbook. It's that integrin protein that's important for that connection. And this leads into the next topic, which are these cell junctions. So how are cells connected to one another? Here we see this cell connected to that basal lamina through integrins, but they're also connected to one another. So neighboring cells are adherent. That gives tissues their shape and structure. You know, we don't just have cells loosely floating around in a lot of our tissues, but they are connected, and they're connected in a very certain way. So let's start with plant cells and these interesting connections that you find there, and then we'll get into the animal cells. So a kind of connection we see in plant cells are called the plasmodesmata. And remember, we've got these cell walls, these thick cell walls that prevent uptake and loss of water, but that creates a challenge for moving material from one cell to the other. And so how plants do this, they actually have little pores that connect different cells. In a way, you can think these cells, actually the cytoplasm of these cells is connected. So it's continuous from one cell to another through these channels. All right, how about animal cells? So your textbook gives 3 examples of junctions, and I'll throw in a couple extra ones here. But you can see the 3 from your book here. There's things called tight junctions, desmosomes, and these gap junctions. So let's look at these a little more closely here. So here's an example of a tight junction. The red here are actin filaments, so they're kind of tied into these tight junctions, but you have actually other proteins that are making the junction itself. So here's cell membrane from one cell, here's another cell membrane, and these claudin or occludin proteins connect the 2 cells. So this provides a pretty tight association, hence the name. And if we go back to our intestinal epithelial, so that's really important because think of all the stuff that's moving around in your gut here. It's important that material doesn't squeeze between the cells and get elsewhere in your body, right? We want whatever is in our intestine to stay there. And so these tight junctions prevent leakage from happening between those cells. This next one here, these are called desmosomes. These connect to those intermediate filaments. So think back to that cartoon of the skin layer where you have all these connections between cells. A lot of desmosomes in skin cells. These cells, the cells themselves are connected by cadherins. So these are proteins that are on one and the other cell, and then they connect in the middle. So this isn't like a tight junction. There's actually space here, but this is a pretty solid connection between those 2 cells. All right, the other one from your book here is the gap junction. So here we actually have pores. So these proteins form a channel in the cell membrane of one cell and the other, and then they connect in between. And so this creates a channel. And so here we see a little molecules here passing from one to the other. These could be really important in, say, cardiac muscle, for example, when there's that electrical potential through changes in ion concentration, where it's happening in one cell, but because there's direct connections with the neighboring cells, that message happens quickly for those cells too. So it happens to a group of cells almost instantaneously because of these gap junctions. All right, the bonus ones here, so to speak, your current textbooks kind of skips these over, but the adherins junction, these guys are connected through cadherin proteins as well. We saw these in the desmosomes. So it's just connecting one cell to the other. But on the other side of it here are actin filaments. And so these are these bands of actin filaments that might be important for providing structure and connection between two different cells. But the actual connection itself are the cadherins. And the other one we see are these hemidesmosomes. So it's like a desmosome, except we're not connecting cell to cell here, but we're connecting cell to something else, extracellular matrix. So like the basal lamina, and that's that integrin protein yet again that's making that connection to extracellular matrix. And on the other side, connected to intermediate filaments. All right, let's see how you do. The plasmodesmata in plant cells are similar to what kind of connection that I just talked about in animal cells? All right, yeah, it's the gap junction. So that creates this direct connection of the cytoplasm from one cell to the neighboring cell. And so that's how those are similar. All right, so let's just bring this back around to some of our bigger, broader themes here. This idea, we've talked about the individual pieces and parts of cells in this chapter, but we got to bring it back to that idea of emergent properties. So the cell is greater than the sum of its parts, right? So these components we talked about, they aren't working alone. They interact with one another. In this little cartoon that they post in your textbook here, here's a macrophage catching bacteria. And what's happening, those cytoskeletal rearrangements, lysosomes are going to be important for digesting any bacteria that are taken up. Gene expression happening in the nucleus is going to be important for dealing with those broken up pieces of bacteria, breaking them down metabolically with the mitochondrion. So all these things are connected. And if you didn't review the slides from lecture 1, the ones that I posted, but we didn't have time to go over in class, here's this idea of emergent properties. Again, this idea that the sum is greater than just the individual parts, right? So take a car, right? If you take all the parts and look at them individually, that's all very interesting, of course. But this jumble of stuff doesn't do much. It's not until you put them together, not just put them together, but put them together in a very specific way that you get something that has emergent properties. This thing can move around, right? You can go places with it. All this jumble of stuff, you can't do that. So all of a sudden you have something emerge at this next level of organization. All right, so think about that. And we'll end here with a final question just to gauge your understanding. And we'll see you next time in Chapter 5.